last time, but I wasn't able to do the stream because I think it did some weird host bounce back.
Okay.
But that's okay now.
Yeah,
And
I just maybe we can wait 2 minutes. Yeah,
yeah, no, that's fine.
yeah, yeah.
But in the end, did you get the recording for
Yeah,
the
I've got the
okay.
recording. I had to cut off the first 10 minutes because it was just all of us just faffing around. Um, but yeah, it's fine. And then
Okay.
I'll get the recording uploaded hopefully today.
Okay. Well, I think that maybe we can start because I still have a brief introduce to these two sessions today.
Okay,
And
I'll
the
just
first
start the
section is
recording. I'll just start the recording now.
okay. Yeah. Yeah.
There you go.
Okay. So the first section today is still the genomics and informatics tools and database. And we're happy to invite the Dr. Matthew from the Sanger Institute and Dr. Amelia to introduce the BGP portal is a framework to maybe get suitable to many other kinds of genomics projects to store and visualize the tools and the data. And the third part is Dr. Terence will show some the NCBI toolkit, which the power and to show the genomes data. And the second sections we will change to another Zoom link and we'll still focus on the data and the IT part in genomics. And the Juan Pablo, Dr. Juan Pablo will show the EBIs about diversity portals. And will the Dr. Zhen Li from the Ghent University will show some um, just some duplications, identification uh, ideas about the genomics data and the whole genomes uh, data analysis. And finally, the Jerry Wang from National Gene Bank, he will show some ideas about the system he's uh, under development. So so we just start because I think we can leave more attendees to have some question time. And the first show is from Dr. Matthew, and very happy to invite him to have a presentation here. And right now he's the leader and of the informatics infrastructure teams of the Tree of Life program, and he has a very great experience in the genome assembly pipelines, and right now is have some these tools already used in some large scale genome analysis. And also he is also the used to be the team lead of the examples teams. So I think today he will some his ideas and he designed in his work now.
Thank you for the introduction. sure thing. I'll share my screen now.
Okay.
Just to be slightly more accurate, I was leading the comparative genomics team of Ensemble, not all of Ensemble. Still be happy about that. Can you see my slides? That okay, super. All right. So yeah, so now I'm at the Sanger Institute in the UK, in the Tree of Life department, where I lead the informatics infrastructure team. And in that role, we have are defining standards for writing all bioinformatics pipelines. So I've got a few slides to introduce what the program does at Sanger. Then we'll talk about what those standards are, and then some thoughts about how to connect it and how to take it forward in your own, for your own software, own pipelines. So we're a scientific program and department of the Sanger Institute. It's called Tree of Life. And we're actually focusing on non-human sequencing because we're sequencing biodiversity at large scale, very, very large scale. But it's not just for the sake of it. It's that because we know that understanding how biology, how biodiversity works, will help us design new drugs or find new insight into genomics, and I will eventually know better human condition. That's the purpose of the program. So we got the whole range of projects doing whole genome sequencing, can be in specific region of the world or specific taxa, specific ecosystems. And we're following this very simple motto from Mark Blackster. He just wants to sequence everything. And that's what we do really. So we are involved in a project that you may have heard of, the Darwin Tree of Life project. It's very well funded and it's about sequencing all species in the UK, eukaryotic species. And it's not the first project. I think before that, the Vertebrate Genome Project had a very good go. And then there's been a lot of structure being created on top of that. So now the hierarchy is that Darwin Tree of Life is part of the European French Genome Atlas, so ERGA BGE, actually. That's focusing on Europe, but it's all under the big umbrella of the Earth Biogenome Project. On Earth, there are more than between 2 and 3 million species, perhaps, and that's the ultimate goal. Right now, in our department, we have, I think, just sequenced about 2,000 species, so making a high quality reference genome assembly. That's across a variety of projects, specifically about 1,500 for Darwin Tree of Life, 200 for VGP, and then we've got a few hundred for the ASG project, which focuses on marine systems. That just gives you an overview of how broad our endeavor is. So to sequence a species, it's a complex chain of people working together, starting with sampling species in the field, sending them to Sanger for DNA extraction sequencing. My interest is on the informatics part. So everything that starts here from genome assembly, and because that's where we've got the informatics pipelines happening. We're organized in 3 teams. So first, there's the assembly team who do the QC of the sequencing data and make a draft assembly. In the assembly curation, we'll curate the assembly and submit it to ENA. And then my team enters and we run the analysis pipeline on published assembly. Overall, it's about pipeline that will take genomes in and will generate some data out. But we want to make it in a way that it really serves a higher purpose, and it can be beneficial for everyone in the biodiversity genomics community. So our pipelines, overall, we're on pipelines to import and pre-process data, do some automated genome assembly, check for contamination, help the curation team, and really make a top quality genome sequence. And then following the best practices of scientific publishing. For each genome, we generate a publication called the genome note, which needs its own set of analysis and reports. But it's part of a bigger project called Genome as a Party, for which I will have a talk at the Biodiversity Genomics conference in a few weeks' time, which is about running a lot of analysis and sharing that to make it sustainable. So, overview of how we're doing things, and I will dive into those topics in the following few slides. We decided to use Nextflow. We follow the NF core principles. All the software is public on GitHub, open source with the MIT license. And then all our pipelines are versioned and released in Zenodo. That's where they are accessioned, get a DOI. So let's dive a little bit. Nextflow. So Nextflow is a workflow management in a workflow language. So that's how language in which we describe which steps, which software to run in which order. It's a very popular one. It had a large user base at the Sanger, but outside as well. One really key element is that the company behind Nextflow, Sekira, they do listen to their users. So they've got big community on Slack. They have regular events where we can meet them. They come to the UK, they come to their countries. We go there to attend the big summit they have every 6 months. So there's that communication going on. And even though Sanger is paying for an official support contract with Sekira, actually we don't really use that often because we interact with Nextflow folks via the community, and it's very efficient way of doing. So they've got public documentation, training materials. They got regular training events. So that's how we can onboard new developers. Nextflow as a workflow system, it has large support for different HPC and cloud environments. So that tells us that if we develop a pipeline the right way, the right framework, which I will explain next slide, then our pipelines can work out of the box in different people environments. We know Nextflow can cope with very large scale analysis and has a very nifty graphical interface with it. But Nextflow is just the workflow language, a bit like C or C++. You need something on top of that tell you how to write good quality code. And that's where NF core enters the scene. So it's a public set of standards, guidelines, that will tell how to develop pipeline, how to include documentation, configuration, how to help separate those things, how to format your repository. So all of that, just essentially following what the NF core say. We didn't need to write our own standard, we didn't need to diverge. So it simplifies things, communication with the community, because anyone there who's familiar with NF core can use our pipelines and vice versa. And they developed a lot of tooling around that. So maybe instead of saying C++, I should have said Python. Next is like Python, it's just a programming language. The NF core environment will mandate a way of printing your code, writing it. That's a way of packaging. So it will stuff include how you run Black, how you run your package installation, package management, all of that via the command line tool. Then it also tells you how to structure your pipeline. So they've got this concept called module, and they're very tiny pieces of Nextflow code. And there's a one-to-one correspondence between one module and one software. So you got one for some tools, one for Minimap, and so forth. And then when you write pipeline, you combine this module into sub-workflow. And sub-workflow, you can combine them. And as you go up the chain, you end up with your big workflow that addresses the question you want to answer. So at the bottom of the pile, we've got those modules. And very importantly, they all refer to software via bioconda and biocontainers. So it means that all the software dependencies are very well versioned and containerized. And when I say that Nextflow has got a large user base, NF core even larger. On the Slack workspace, they've got more than 10,000 people. So the way we've done it, we've got our GitHub space. we decided to go very easy on license, just MIT, everything. So it's open source. Anyone can use and modify the pipelines. All the code is on GitHub. Most development is done on via pull requests and on public reviews. So the usual pull request welcome, and we really mean it. So there's been a lot of interactions with users who would report issues or suggest code changes, and all of that can go through publicly, and that's how we can improve our pipelines. Together, we try to define a nice test data set that our pipelines can at least be tried by people in different environments. And I've got a link to that in a few slides. One very important thing is that we can link to Zenodo. Zenodo is a place where we can deposit digital objects, and they can be accessioned. So it's used for many things like presentations and so forth. But the link with GitHub means that whenever we tag a new release on GitHub, it makes a snapshot to Zenodo. So that piece of code is now set in stone in a different host than GitHub. So even if GitHub goes down for whatever reason, we will still have Zenodo. We will still be able to access our software, and it's accessioned. So now there's the accession number, which has a list of authors and contributors. So there's a way of crediting as well everyone who's involved in software. Where is my cursor? Yes. We have shamelessly forked the NFCore website, and oh, it's going too fast now. Sorry, my computer is doing funny. It's very spooky. It's Halloween. Okay, sweet. Right, so this website, I think I see, okay, you will see it's a fork of the NFCore website. We've just changed the colors and the text. But at least we've got now have a space where we can list all our pipelines and the standard way of showing what all pipelines can do, parameters they use, the outputs files, and then some more documentation there. We also can expand that to tag any other documentation about our processes and software we use. Okay. At the time, we've got about 15 pipelines in development at different stages. 9 of them have reached version 1.0, 2 are very close to it. And we've got 6 that are used daily in production to generate those generate assemblies and those analyses I was talking about.
Sorry, Matthew. It keeps flashing black.
Oh.
I don't know whether your computer is on its last legs or something, but the share screen keeps flashing black.
Yeah.
Okay, let me stop sharing now.
You can share again.
Plugged into the power, so there shouldn't be a power issue. Okay. I can share again. Let's see if it works.
Yeah.
No, I think it's my Chrome. Chrome is behaving, it's a bit naughty now. I'm not sure why. Agent responsive, that doesn't help. What do you see now?
This is your Chrome page.
Okay, let's try slideshow. It is still loading.
It is, yeah.
It's a dark screen.
Okay. I don't know what happened. I'm sorry about that.
Can you shift and see if you can display the portal page itself, because it might be the Google Slides thing instead.
Yes.
Or maybe if you get out of the full screen mode.
Try to shut down Chrome. It's just very unresponsive now. Sorry about that. Um, okay, now can we start it? I think what was happening is that I was in a power source that was not powerful enough. So my battery was still depleting and was uh, close to the end. I changed the power source and now Chrome looks much happier. Okay, so I think it feels much better now. All right. Okay, we lost time, so I tried to go quickly. There was a BGA last week about Genomic pipeline, so it's a public demo. You can go there. There's the link, there's YouTube, everything. What's important is that you can run the pipeline. Anyone can run the pipeline with those test data that are provided within it. So the key there, and that's what I want to highlight the last part of my talk is that this pipeline takes a FASTA file. You can see it's referenced by, so we take it on the web. It's referenced by accession number there. It takes some biosamples, accession numbers here. And then the input files are regular BAM or CRAM files. So I want to talk about this publication from about 2 years ago about the 5 principles for research software. So the 5 principles were designed for data initially, but this is a slight adaptation to work with software and dependencies and all of that. I think it's really key to make the pipelines usable for the community. So for instance, is findable is there a standard place where you can find easily the software, both for humans and machines? So no, our take on it is version the pipeline software, every important release, and make it and accession it on Zenodo. There's also Workflow Hub as a very good alternative. And both platforms offer forms to enter a lot of metadata about the software. Workflow Hub has got a concept. You can really name all the authors and tell how they grouped into teams, into research projects. So that means that you can then easily link things, understand how it's all related, and find that in a standard common place. Accessible. Well, actually, if you follow the first step, then naturally Zenodo and Workflow Hub will give you an easy way of accessing all your releases. It's all standard and accessible. But I would say as well for development, use a public forge, GitHub or GitLab, whichever you like. But it just makes it much easier to interact with the community, with the users, because inevitably you will want to communicate and have some feedback. Interoperable. So it's about how your software works with inputs, for instance. So the Genomic pipeline, we could have made a version that hooks into Sanger systems, but it wouldn't have been useful for anyone else. So our design decision was to make it use input files, output files, and all in standard file formats, FASTA, BAM, CRAM. And then it means that anyone can use it because it's all easy and straightforward. And then if you want to integrate that with our Sanger system, we can still, if you want to. And then similarly, when we refer to external data, use public identifiers like accession numbers to make it clear what the data are and where they come from, and tap directly into the source, like in ENA if possible. And finally, reproducible, reusable. So it's more about like our tech is to cite all software that are used in the pipeline and pin their version, their dependencies. Software containers are a great way of doing that. It ensures that even if the source software goes down, you still have one compiled version available that you can use to run the pipeline. Semantic versioning is also extremely useful because it tells your users how your pipeline changes and whether it is still compatible or not. So, sorry for the rushing at the end. This is my summary. What I said, Nextflow and NFCore, we're happy with that. Open source development on GitHub, Zendo, and very importantly, FAIR principles. So is everyone part of the three teams I mentioned? Thank you, everyone. And then if you've got any question, I'm happy to hear that. I'll stop sharing.
So any questions? So I just thinking that just sharing the fair principle, and because right now in National Gene Bank because we have a bio samples repository, and we also have a sequencing platforms like I think Sanger also have sequencing platform, sequencing machines, and we right now we have a database we call it CNGBDB, but many times that we are still right now we use the workflow language WDL, to develop the workflows. But how do you think that if we can, because right now we not just the the biodiversity research, but also some human genetic research in National Gene Bank. But we still want to run a system, just that the system once that the data is produced by the sequencing machines, it can directly or automatically analyze it by all the pipelines. So how do you think that we still need to develop such kinds of system, or we just let the bioinformatics researchers just use the pipelines and the script? Yeah.
So that reminds me how I think I would say CWA was created. When I meant the leader at the time, that was back in 2015, I think. The main goal was for people to move away from shell scripts and allow them to organize their pipelines in a more standard format, right? And that does the same purpose as SnagMeg, Nextflow, all of that. I think now I would consider that to be the basic way that every bioinformatician should be doing. Use a standard, I mean, a commonly used, commonly known workflow language. But then that, like you said, it shifts the focus onto the support, the informatics or infrastructure team, how to integrate that into the environment. Because a lot of, I've heard a lot of IT departments who have constraints about what software, what workflow manager, how you can run things. And that's usually something to take into account. So we are lucky at the Sanger that there were not really such restrictions. So we were quite free to choose the workflow manager that we liked. But I'm aware that other institutes may not be in the same position. So it's part of it is probably coming through having a standard or some standard document that's promoted by an organization like the EBP that we can then show to decision makers in institutes to tell them, okay, let's say they don't like SnakeMake, but we can show them, well, SnakeMake is not an esoteric thing. It's a very commonly used workflow manager, right? But if there can be like a standard document, a policy that we can refer to that can help them convince the administration team. As a thing, not to make it that I'm an explore-only person. We need to analyze that they're all good systems. They're all good on their own right. We just chose one and we follow.
Okay. Thank you, Dr. Matthew. So yeah, yeah. So happy that Matthew can share his experience and story about these parts. I think in futures that in many projects in EBP we can share much more ideas about the standards pipelines or some standard workflow language about this about the change. So the next speaker is from the Center of the Genomics Regulations, Dr. Emilio, and he's informaticians at CRG, and he's focused on the API specifications and software development, and some data management, and you also have a project called the BioGenome Portal, and it's web-based applications and to facilitate the organizations and access the data. So right now you also have a best practice of the this portal. It has a practice and take as a reference portals and the BioGenome project. So thank you.
Hello?
Can you hear my voice?
Yes.
Hello?
Hi there.
Nice
Can you hear
to
me?
see you again.
Nice to see
Yes.
you.
So
Nice. Let me share
okay.
this.
So okay.
Okay, cool. I guess you can see my screen now. Or maybe still loading.
Yeah, we can see your screen.
Cool. Nice.
You just show, yeah.
So welcome everybody. I'm Emilio Righi. Actually, I'm a full stack developer working at the Center for Genomic Regulation, but I'm also involved in the Catalan initiative for the Herb Biogenome project, which is again another biodiversity genomic project initiative that aims to sequence all the species under the Catalan-speaking territories, which are like Catalonia here, Barcelona, but as well Andorra, the Balearic Islands, and the Valencian community. So as we know, there are many types of biodiversity genomic projects. Some of them are focused in catalog and sequence species in specific regions, such as DITOR or as well the CBP itself. Some of them want target like specific groups, such the Vertebrate Genome Project or the B10K, but as well as some are like global efforts, such as the Herb Genome Project, or some are conservation focused, like the California Conservation, or they aim to model organisms, like they focus their research on model organisms, like the USCS Genome Browser Project or GeneCode itself, or some of them as well target ecosystems. But we can say that all of them have common challenges, such as data management, public engagement, and interdisciplinary collaboration. And that's why in my lab, we have developed the Biogenome Portal, which is a web-based API-centered platform designed to manage and track data generated by biodiversity genome projects. So here are some of the features that the Biogenome Portal or BGP have. But the main important, and I will talk about this feature in the next slides, but the main important thing is that this portal is possible to use it everywhere. It can be locally or on private networks on the internet, depending on the use case. So the data integration in the Biogenome Portal works like the following. We have two main sources of data coming from INSDC and data coming from the users. Both of these data are linked to a set of NCBI taxonomic information in a relationship many to one. INSDC data consists of the following. Assemblies are imported either via assembly or by project accession through the NCBI dataset v2, the latest version of the NCBI dataset API. Experiments are as well imported either via experiment or by project accession, but this is done through the ENA portal API. And while biosamples are imported either via biosample accession or project name. And this is done through the biosamples API. While on the other side, users can import local samples in the form of spreadsheets, like sample metadata spreadsheets. Then they can import as well, upload GFF files and link them to the assemblies imported from NSDC, but as well they can handle GOT reports, GOT compliant reports in our portal, and they can as well enrich organism metadata, so this set of taxonomic information, with publication links, vernacular names, images, and as well custom fields. So as I said, our portal, the BGP, is able as well to manage GOT compliance reports. Indeed, users can upload and download reports either via API or via the user interface. Depending on the user permission, they can like upload species which they are authorized, like assigned to them. But also they can create new species that are not present in the portal. And then once all these users, like our collaborators, upload the GOT status, the GOT reports in our instance, the portal itself is able to generate reports that meet this GOT standard, like customizing the header of the GOT report depending on the project. Indeed, in the CBP data portal instance, we are actually using these features to generate and submit GOT reports directly to GOT. Indeed, GOT is like syncing with us because they are consuming our API. And so in this case, it's possible like the user updates the GOT sequencing status of their single species through forms or TSV uploads. Then another feature of the portal is that it's able to track the NSDC submission progress of a specific by-project accession, thus automating data retrieval and updates to provide accurate submission status for each organism. So we can configure scheduled jobs to retrieve assemblies, experiments, and biosamples and storing the new records. And then each organism in NSDC submission status will be based on the associated data with it. Then the BGP provides as well powerful data analysis and export capabilities, thus enabling users to filter, visualize, and export metadata with ease. Indeed, it is possible to apply personalized filters to refine metadata searches, as well to then download the filtered results in TSV formats, and create and download tailored charts based on the filtered data. We will see that in a moment in the demo. Another feature is that the BGP standardizes geographic location data. Let's say that we have like the important metadata from spreadsheets, so by the users or from biosample, are converted in a common format, and then the BGP itself verifies if this set of coordinates fall within some country boundaries. And then a part of all these features, the BGP is very configurable. Indeed, this is possible to configure almost all the user interface, like the pages we want to display, the charts in each page, and as well the filters that are used to query the database. Then there are some scheduled jobs that can be configured or triggered via API, like the INSDC submission tracking that I mentioned earlier. And then as well, it is possible like to use specific taxonomic identifier as root nodes to focus only on data relevant under a specific taxonomic group. And then as well, it is possible to upload any type of sample metadata spreadsheet. To further, let's say, control the collaboration between the researchers, there are two main types of users in the Biogenome portal. One is the admin, which has full access to all the CMS. It says, let's say that it have all the CRUD actions enabled, and is as well authorized to create and manage data manager users, and then can assign specific species to these data managers. While data manager has access only to the species assigned by the admin, but can as well they import new species and assign the imported species will be assigned to themselves, and only if the species is not present in the database. On the other way, it will be unauthorized to access the species or manage the species. Right now, we have 4 active instances online on the BGP. The first one is the data portal of the Catalan initiative for the Herbal Genome Project, which was like, let's say, the main use case to develop this software. And it tracks all the data published to NSDC under the CBP umbrella, But as well, there are some curated data, such as the GOAT sequencing status, some species photos, vernacular names, publication, local samples, and I forgot to mention here as well genomic annotations. Then we have three kind of examples to show like the flexibility, but as well the possibility of the BGP to adapt to the various needs of the biodiversity genomics projects. One, like that tracks all the data published to NSDC under the ERG umbrella. And the other one to further show as well the scalability of the software and how the software can handle large datasets, that is tracking the NSDC. That is tracking the data published to NSDC under the EBP umbrella. And then we have as well another example that contains all the specimen of a lichen herbarium, and this dataset has been retrieved from GBIF. So now I will super fast show you a little demo. I think I'm in time. I think I have 4 minutes more. So can you see the screen now? Can you hear me?
Yeah. Yes.
Yes. Okay. Sorry. So here we have the official data portal of the CBP. It is like we can select the different languages, and as we can see here, we have all the things that I've mentioned, like all the biosamples imported under the CBP, as well all the experiments linked to the CBP, and as well all the assemblies. And then we have as well the organism. As we can see here, there are this set of taxonomic information that came from NCBI, and then many of these statuses have been like manually updated by the users which this species is assigned to. And then from here, we can see all the data related to this species, like this metadata that has been added by the users, the annotation that has been uploaded by the users. We can as well download the annotations in the various formats, and we can see the source of the annotation, all the related assemblies, the map with the collected species, the images, the vernacular names, if any, and the publication links. Then we have as well here the link to the GOAT report. It means that by clicking here, we will retrieve our GOAT report, which is the one that actually updates the GOAT website. And then here I am already logged in. We have our admin areas where we can upload the samples or upload a GOAT report to manage the users. In this case, I'm an admin. Or we have our local samples. We can create a new organism starting from the NCBI taxonomic identifier, or as well we can upload annotations in GFF format, or link or add some links to the annotations. And then here we can actually, I will show you this use case. Here we have, for instance, the assembly of Sirictis novacula. We have uploaded two different annotations, one coming from NCBI and the other one coming from Ensemble. And actually, the BGP have a genome browser, which is a React component coming from the JBrowse community. Actually, my stack is Vue, but I somehow managed to use a React component within the Vue framework, which are two different kind of JavaScript frameworks. And here, as you can see, we can explore the annotation that we have uploaded. And then we have as well the sample map, where we can filter by sample or sample type in case if there is any local sample in this application in the BGP instance. And I think from this side is almost everything. But then we have as well this taxonomic explorer, which is we are using a D3 hyperbolic tree to explore the taxonomic tree of all the data generated under the CBP. So here, as you can see, we have like the Wikipedia description of Eukaryota, but I don't know, maybe there are some aves in the CBP. We go to aves and we automatically are redirected to the aves. And then we can see as well all the related data to aves with the map, their sample location, the Wikipedia, and we can change the language, and the Wikipedia will like reactively change depending on the language. And yes, and then here there are some charts but I think it's way more cool if I show you this feature in the example instance that is collecting all the CBP, or sorry, all the Her Biogenome project data. So this instance, as I said, is like tracking all the data that are being generated under the bioproject accession of the EBP, of the Earth Biogenome project. And here, as you can see, there are many more data. For instance, there are like 6,018 assemblies, and we can like see the submission, the release date trend of these assemblies, and as well some other statistics. And then from here we can like filter for EBP metrics and how many of these assemblies like comply with the EBP metrics, or if the blob toolkit ID exists, and so on. And these filters have been like configured before launching the hub. It means that here we can configure whatever we want, as well as the column. I've decided which column to show, and each instance can be like modified and configured depending on the use needs. And so we can, as I said earlier, we can like see the different, like the reactivity of the charts depending on what we, you know, on what we query. And as well, then we can export our query results in a TSV file, and we can as well choose which columns we want in the TSV file, and we will have here our TSV file with the like the full columns, but we can filter by whatever column. And as well, we can create like custom charts. For instance, this is due to the fact that the metadata sometimes are different from like biosamples coming or submitted in NCBI and biosamples submitted in ENA. They use like different checklists, as we can see here. If I'm not mistaken, this has been submitted from NCBI. Yeah, this is like the NCBI checklist of the biosample, and this one is the ENA. So depending on that, we can like filter different metadata. For instance, if I want to retrieve all the, let's see, like this one, when all the biosamples contained in Eukaryota under the Her Biogenome project have been released or published under the NSDC, I will go here. As this is a metadata field, I will like write metadata point, but is this, the description is here, and then I can choose between different charts, and I will broke the portal as always. No, I'm kidding, but yes. And here we can see like the release trend of this metadata field, but we can do it with any kind of metadata that we find interesting, and then we are able to download the generated charts. I don't know, sorry, no, it's not adult, but it's live stage. If we go here and we live stage and we want, for instance, a bar chart, we will submit and we will generate a bar chart. Or we can filter here, and we can do pretty much everything with this, with the things we have here. And as well, again, we can like filter our research by a taxonomic ID, and then again we can keep filtering. And in this case, as I mentioned in the presentation, we have as well a map containing all the species which samples have been collected within the boundaries of a country. For instance, we can see that, and from here we can see like all the, sorry, there is some problem because the bit, okay. And we can see all the species collected within that country. This does not mean that the species is actually being sequenced by the country, but this just means that the biosample related to the species has been collected within the boundaries of the country. And so that's it from my side. I don't know if you have some questions. Here are some useful links if you are more interested in the project, or I'm more than happy to help you launching the instance or explaining whatever you need.
Okay, thank you, Amelia. Yeah. Any question for this? Yeah, I think you are really a full stack developer. There is not much stack developer in BGI. Yeah.
That's true. But I actually do work as
Because
well
the
with
whole
Nexus pipelines. So.
I have a quick question. I don't know if we have enough time for them. I think the session's supposed to end in 10 minutes, so I'll be quick.
Mhm.
I was just wondering if internally I'm using the portal as my project tracking system.
Mhm.
Is it easy to create a field where I have my species name, my taxon name only, and then the status internally until it gets a taxon ID approved?
No, right now, no. Because we actually use the taxonomic. I think it's the same problem we got
Yeah.
no? And everyone has this problem because, of course, we use the taxonomic identifier actually as the unique identifier of the species. So, no, but what I thought about that because we have a metagenomics project in our lab. And so what you can actually do is create a record, but within the closest taxon you have. And then you will just add, samples to that. You will link samples to the taxon, and then you will switch the samples again. But it's super easy actually because you can download the TSV and then update the tax ID and upload the tax ID TSV.
That makes sense. It'd be really nice if we had this type of service as one of instead of a Google Sheet, like most of the projects are doing nowadays, just have a system that is all kind of set for them, and all they have to do is just include the species and the status, and then we can just exchange information between all the portals.
Yeah,
this is something that we should be discussing, I guess.
Indeed. But as well, what I was trying to create was like exploiting the GitHub projects and try to automatize like that users, but then I leave it because it was too complicated for me. But like creating issue specifics with some templates, and then the user can update their species there, and then you can somehow create a webhook or whatever that generate the TSV within the same repo. That would be super nice because at least you don't have to manually, go get all the Google spreadsheets because sometimes it's painful as well for the eyes.
Yeah, I guess we are getting to the point where we need things that are more user friendly for the general people that are kind of interested in the species itself and the sequencing.
Yeah, that's why what I would talk was like just everybody gets his own GOT report, but about each species, up to 9, 10 species, I don't know. And then they will upload these GOT reports in our portal, and then the portal itself will generate the GOT report without like taking care of hundreds of columns. This is not our case for the moment, but will be. Sorry, I'm taking, I guess, too much time. I'm
amazing. Sorry, I will stop too.
Yeah. So thank you, Emilio. So
Thank you.
we move to the next. We need to move to the next presentation, and very happy to invite Dr. Terence from NCPI. Well, right now is focused on the project called CGR, is short for Comparative Genomics Resources, and it has some development work for the reference sake about this part, and his teams also developed some methods for genome annotations and the eukaryote and tools of identification of the sequence contaminase and all of these new existing genomics. So welcome, Dr. Terence.
Thanks.
Yeah, can you hear my voice? Yeah, thanks.
I can hear you. Currently somebody's sharing and I can't steal the screen.
Okay.
So if whoever's sharing right now could stop.
Okay.
Great. And let me get my slides up here.
Yes, okay.
Great. So you have the right view? Yeah, so thanks for this opportunity to talk about what we've been working on here. So I'm at the NCBI. I'm going to talk about this project we have called the Comparative Genomics Resource, which is really about trying to better support comparative genomics in eukaryotes. So I'm going to give you a little bit of a background of what we're trying to accomplish. I always like to start here. Seen variations of this before. This is just showing the influx of new genomes coming in each year to the INSDC. And just looking at last year alone, we received over 9,000 genomes, of which over 4,000 were the first genomes available for that species. And overall, we're currently at over 44,000 genomes for eukaryotes from over 18,000 species. And we know this is all just the tip of the iceberg with ambitions for EBP and all the related projects out there, aiming for hundreds of thousands and even millions of genomes in the future. Just being able to talk about growth and scope like that, I think, just like to emphasize, this really emphasizes the value of the INSDC, the International Nucleotide Sequence Database Consortium, that's made up of DDBJ and ENA in Europe and GenBank and SRA here at NCBI. So these are the central archives of sequence data, raw read data, and all of the associated metadata that goes with them, taxonomy, bioproject, biosample, the assemblies, and linking out to the literature. And I hope it's a given with this audience how valuable INSDC has really been to all of the topics that are getting discussed here, with key emphasis on things like unique identifiers, accessioning for all of these objects, common data standards, open and unrestricted access, global coverage and accessibility, and my completely unbiased opinion, I think INSDC Archives is probably the most successful scientific database of record out there. I think it's really amazing what's been achieved by having this common archival repository. But as successful as INSDC's been, the goal is really not just about archiving data, but it's to help the scientific community making use of that data, making new discoveries. And on that end, we've long recognized there's some foundational problems to making this work. One is this issue of exponential data growth. More data is great, but there's issues with data quality and accessibility. Another is all of this data is kind of spread all over the place. Just even within NCBI, we have different databases, and for the sequence data itself versus particular metadata. So that complicates accessibility. Then there's outside resources that also have valuable information, but you kind of have to know what is available where, what organisms are supported in different places, and there's really just a pretty small number of organisms that have really deep support. So you have to go hunting around. That complicates use. You have to know where to go. You have to work with different interfaces. You have to download all of this data. All of that really hampers working with data at scale, which is really the future that we're facing with everything that's coming in. So those are the goals or the issues that led to this project that we now call the Comparative Genomics Resource. And the central tenet here is moving beyond a concept of just model organisms to supporting the full breadth of eukaryote research organisms, going from dozens of organisms to thousands, focus on genomic and associated data, an eye towards supporting biomedical research, but really, it's the whole gamut of biodiversity studies that are really targeted here by CGR. And to achieve this, we have kind of two angles. One is what we can do at NCBI, focus on improving data quality, providing tools, providing interfaces to access data, and also working with communities for improving data exchange, improving connectivity. The goal here is really to kind of build out an NCBI supported ecosystem combined with community resources that together really power this future of comparative genomics. And some of the things that have come out of CGR have been talked about already in previous talks. So we're already achieving some of this global community network here. So I'll give you a whirlwind tour of some of the things we worked on. One of the first things we did was working on better contamination screening to try and improve data quality. And this is something that we've always been very conscious of, because we find about a third of submitted genomes have some level of contamination, sometimes just a little, sometimes quite a lot. And so what we did with as part of CGR, we totally rebuilt our contamination screening system to be much more sensitive, much faster. It's available in GitHub. You can run it in Galaxy, and we're able to go ahead and run this on all existing eukaryote and prokaryote assemblies, over 2.5 million assemblies, with data reports now available on FTP. We're working to make that even more available. The other issue we see on the data side is less than 20% of eukaryote genomes come in with gene annotation into INSDC. So it's just over 15%. And unannotated genomes are really much less useful than knowing what the gene content is. So here at NCBI, as part of the RefSeq project, we've worked on annotation for a really long time. And what we're doing now is turning our eGAP annotation pipeline into a tool that you can run yourself. It's built in Nextflow, uses a Docker container. It'll run in the cloud. It'll run on your local HPC. Really, the goals here are making it really easy to use, high quality, and produce output that is ready for submission to GenBank. We have an alpha up on GitHub already, already seeing lots of interest in getting this going and pushing this forward. On the side of working with data, one of the big things we did as the project is this new data portal called NCBI Datasets, which was mentioned in the previous talk even. The goal here with datasets is really just a complete rethink of how to access gene and genome data at NCBI. It has easy to use web and programmatic interfaces, and whichever interface you're using, you get access to the same data, great documentation, and this has been out for a while. If you've come to NCBI in the last year or so, you've probably already been exposed to this. Nice table views here, accessing over 7,000 genomes under the EBP bioproject umbrella, taxonomy level views, assembly level views, and on all of these, you see easy access to download the data. And that's one of the things that's really been emphasized as part of this datasets project, to not just make it easy to download, but it also synthesizes and combines data from multiple locations, from what was previously across assembly and gene and biosample and bioproject and the sequence databases. Combine that all into a single package that you can work with, giving you data and file formats that you're readily able to work with, and all of this will work at scales that previously would have really been very tedious to do. Going from working at genome level to working at gene level, we've been expanding what's available in our NCBI gene resource, which now has gene level pages for over 2,000 eukaryote species, been adding connections to other resources, more data about things like expression, adding functional information, including gene ontology in here. And you can also access information about orthologs and other species. This is a data set we currently have for over 1,000 species of vertebrates and arthropods, and keep expanding that. We have web pages that let you explore these sets of orthologs, can view their protein architectures. You can build out protein alignments, download all of the data, and as I said, we're continuing to expand the set of organisms that are available here. And last thing I'll show you is this tool we developed called the Comparative Genome Viewer, which lets you visually see similarities and differences between genomes. So this view is showing you a whole genome comparison between this chimp genome. This is the latest chimp genome from the NHGRI T2T primate project compared to human. So you can see at a global level, translocations and inversions and the like. You can zoom in to smaller genomic regions. And this is where I think it really shines, like this region where there's an insertion that splits the human DRG2 gene into two, actually wind up with two related DRG2-like genes in chimp, plus a bunch of novel insertion genes in here. And this is interesting. This is a region that's actually involved in some developmental disorders. So potentially chimp could be an interesting model for understanding what roles of this region. We add organisms to the CGV resource by request. We're now over 400 species and 1,000 pairs of genome alignments supported in here. So we've really seen great engagement with and interest in using this resource. So that's a really quick sample of what we've been doing under this CGR project. As I said, the goal really here is better supported NCBI with these, better connection between databases, better tools, supporting a wider range of organisms and higher quality data, working to this concept of a federated biodata ecosystem, working with community, helping connect and leverage knowledge wherever it may be, always keeping the end goal here. It's not just about collecting data. The goal is about advancing science, and we're in IH. This is all comes down to trying to improve human health. You can find out more about CGR, Google for NCBI CGR, get to our website. We have this email if you want to contact us. We have feedback buttons on all of these pages. So we're really interested in hearing how things work. We have a monthly mailing list that you can sign up for. And on the mailing list, or reach out to us, or watch on the website. We've been very engaged with users throughout this project. It's been incredibly useful, through surveys and individual interviews, small user groups, and user testing, really to help inform and drive all of this development process. So if you're interested in particular resources and helping to further improve those, we'd love to hear from you. Really cast of hundreds that are involved in this. This work spans a half a dozen product teams at NCBI. Really phenomenal group of people. And I'll take any questions.
Yeah, thank you, Dr. Terrence. Yeah. So any questions? Yeah. Okay. Maybe many people are quite already familiar with NCBI projects.
I see Matthew's trying to talk, but I think he's muted.
Yeah, I've got one question.
There you
okay, yeah. Okay.
In my dream, I would like to have more metadata about provenance added to all the files that are uploaded to NSDC, so that we could see, how each file was generated, and because workflow managers can output that, but it's not yet accepted and integrated with NSDC. What's your view about how NSDC is going, and whether there's a chance of getting that, and what is the other people we need to talk to?
You mean the provenance, like biosample collection and assemblies, how assemblies were made and all of that level of detail, or?
I'm thinking of Arrowcrate and what's his name? In the US, is it Biocompute Data Object? The different standards when you can link objects, data objects, files, and how they were generated by which software, which parameters, and have a full chain that way?
I think there have been some discussions. I think that level of detail, there's it's a little hard to incorporate into some of the current INSDC standards. Some of it is, um, I think the current idea is if we can link out to other resources that are housing all of this. So like, if you have an assembly that this has been most talked about at the genome assembly level. Um, um, so to, the full package of how that assembly was created and what software and parameters and all of that. I think the plan is to add one link to a URL that would go to another resource that would be providing that.
Yeah. In the
But
metadata,
I'm not INSDC myself, so I shouldn't really
yes.
I shouldn't be putting words into their goals and mouth there.
So on the assembly record, good sequencing technology, and that can be used for data mining, so we can find assemblies given certain
Yeah.
technologies. But no, being able to add things like whether it's been created, yes or no, whether which software was it? Is it Verco? Was it polished? Yes or no?
Yeah.
That may be useful to the community, I guess. Maybe just I'm biased, I have to say. Speaking on behalf of everyone.
No, yeah, I'm on your side too. I always like having more data, not less. Um, it Yeah, there are some existing fields where this more gets collected at GenBank than at ENA or DDBJ, but we have a history of collecting the technologies used and the software used for assemblies, and it's actually kind of frustrating. Like even just identifying the set of assemblies that were generated with long reads. Um, I tend to just look for contig N50 over a megabase rather than trying to search for a technique, because um, if somebody's put pack bio in there, you don't quite know what it means. Was it an old assembly where they were just stitching contigs with PacBio, but it wasn't really the backbone versus the other. So it's there's definitely a lot of nuances in interpreting this data. That's um, it, yeah, I think there's a long way to go in terms of adequately capturing that in a way that then is mineable. Um, if there's, as I said, if there's good locations where we can start linking to at least start providing this interconnection, I think that's the place that that we're most likely to start with trying to tackle that.
Thank you.
Yeah, thank you. I also have a question, but we do not have much time, so we need to move to the section 4. I also put the link in the chat. Maybe everyone can get access, copy and access. So see you minutes later. Yeah,
See you there.
see you.